Good morning or good afternoon. I'm Valerie Nunez. I'm a postdoc at NYU. And today I'm going to talk about how double opponent neurons contribute to color perception. Before I start, I'd like to thank the color group for inviting me. And then I'd like to point out that all the work presented today was done under the supervision of Bob Shapley at NYU and Jim Gordon at Hunter College. So I'm going to start right away with our big take-home message, which is that color vision as we know it is pattern dependent. At threshold, color detection is spatially low pass, implicating the contribution of single opponent neurons. However, suprathreshold color perception is different. We change from low pass filtering to responding to color edges. This difference comes from the suprathreshold contribution of double opponent neurons. So suprathreshold color perception is not just about integrating color, it is also linked with form. Double opponent neurons link the perception of suprathreshold color to the spatial layout of the scene. Let's look at some color responding neurons in the primary visual cortex. These 2D sensitivity maps of neurons in the macaque V1 are from the paper by Johnson, Hawken, and Shapley. First, I want to draw your attention to the color coding. Red represents excitation to increments, and blue is excitation to decrements. If we start with the single opponent neuron, it's excited by increases in L-cone contrast and decreases in M-cone contrast. This neuron is cone opponent. If we look at the double opponent neuron, for L-cone stimuli, one side is excited by increments and the other by decrements in cone contrast. This gives us spatial opponency. Then, for M-cone stimuli, one side is excited by increments and the other by decrements in cone contrast. For color luminance double opponent neurons, the increments and decrements are not completely cancelled by one another. The receptive fields are usually calculated by differences of Gaussian, as indicated here, but I'm going to represent them by the approximate shapes of the Gaussian bases. I'm representing the single opponent neuron by a circle indicating that the L plus and M minus fields are coextensive. The double opponent neurons are represented by ellipses with opposite cone polarity to indicate the spatial opponency. And here I want to point out the proportions of each type. So what you can see is that for color responding neurons in V1, 3/4 approximately are double opponent and 1/4 are single opponent. Moving on to threshold color perception, Mullen measured the detection threshold of red, green, and blue-yellow grating patterns to determine that color detection was low pass, i.e. has greatest contrast sensitivity at lowest spatial frequency. The low pass spatial frequency tuning at threshold, indicative of the contribution of single opponent neurons, has also been interpreted by many to extend above threshold. There has been some evidence to suggest this. For example, color assimilation may be explained by averaging color over adjacent areas. Think Munker-White illusion. Here, both circles are white, but on the left, the circle takes on a blue hue due to the adjacent blue stripes, while the circle on the right takes on an orange hue of its adjacent stripes. Here is an example of spatial averaging in art. The image has only a few unique colors, but when I make it smaller, you can see that the averaging over adjacent colors brings the image to life, though you can also see that there are luminance elements in the image too. Another example of averaging color is the halftone coloring used in old 4-color comic books. Here's an equiluminant version, but note how the effect is weak and difficult to see without luminance edges. Next, turning to suprathreshold data, these data are derived from measurements taken from the macaque V1 for red-green sinusoidal gratings by Johnson, Hawken, and Shapley. Schlipperk and Engel observed that single opponent neurons are spatially low pass. Double opponents are spatially band pass, and here they have a peak at around 2 cycles per degree. The single opponent low pass response is mostly influenced by large areas of color and patterns with low spatial frequency. This low pass nature of single opponent neurons indicates large receptive fields and averaging over space. If color perception is also low pass and spatially averaging, this implies that single opponent neurons are responsible for color perception. However, this excludes contributions by double opponent neurons, although at higher spatial frequency, you can see that their responses are dominant. It also excludes research that points to the importance of edges, for example, Krauskopf's work with stabilized edges and filling in. Here is an example of color perception depending on edges. This is an equiluminant version of the Chevroy illusion in the L-M and S color directions. There's an apparent increase in contrast at the inner and outer edges of each color annulus. This edge enhancement is due to lateral inhibition, and that requires spatially opponent receptive fields. However, the single opponent neurons in V1 are coextensive, i.e. there is no spatial opponency, therefore they cannot be responsible for this effect. If you find that the top left circle, which has much smaller change in contrast, is disappearing into the green background, then this is an example of the Krauskopf effect, where the green color, which is next to the white edges, fills in the entire square. Again, the color contrast at the edge is crucial for this effect. Given the color detection is low pass, but the majority of color responsive cells in V1 are band pass, then we ask which give greater responses above threshold, fine color patterns or large colored areas? And can we relate color perception to the action of V1 single and/or double opponent neurons? To do this, we conducted measurements of physiological and perceptual responses using the chromatic visual evoked potential and also saturation ratings. We chose stimuli to isolate responses from single opponent and double opponent neurons. Double opponent neurons do not respond to large areas of color except at the edges. So we created a stimulus with no sharp edges to favor responses from single opponent neurons. Here, the central square fades gradually to the background gray. Then we chose a color complement checkerboard with a spatial frequency high enough that the single opponent response was effectively 0, while the double opponent neuron response was not. There is a central 8x8 checkerboard. It also fades to the background, but it also has high contrast edges. So this stimulus favors responses from double opponent neurons. Lastly, we included a color gray checkerboard of the same spatial frequency as the color complement checkerboard to elicit responses from both types of neurons. Each stimulus was equiluminant with the background, and this was determined for each participant using HFP. Therefore, the results that we obtained were due only to color contrast. Here are the stimulus colors that we used in CIE space, and also in a cone contrast version of the equiluminant plane of DKL space, based on calculations by David Brainerd. The representation of DKL space here is useful because it separates responses from the magno, parvo, and koniocellular pathways along its 3 axes. We only used colors in the equiluminant plane, so the contrast along the third luminance axis was 0. Here, the x-coordinate is the L-M cone contrast, and the y-coordinate is the S-cone contrast. In the L-M direction, L-cone excitation is equal and opposite to M-cone excitation, and the S-cones are not excited compared to the background. In the S-cone isolating direction, L and M cones are not excited compared to the background, and only the S-cones are excited. The L-M color direction was chosen to isolate responses from the parvocellular pathway, and the S color direction was chosen to isolate responses from the physiologically different koniocellular pathway. This was because we wanted to see if the koniocellular pathway would follow the same rules as the parvocellular pathway. In addition, a third color direction, seen here as red, corresponded to the color direction of the red primary of the OLED screen. The red color direction was approximately midway between the L-M and S axis. Therefore, stimuli along the red color direction should produce a combination of responses from both L-M and S-cone driven mechanisms. stimuli were presented at several different cone contrast levels, up to the maximum possible within the screen's gamut for each color direction. The distances along the axis correspond to the L-M or S cone contrast respectively. For the red direction, the contrast of a color is calculated as the Euclidean distance along the red direction using the L-M S axis. The cVEP experiment, participants were adapted to the gray background, then stimuli were presented for half-second periods. This was repeated 30 times for each pattern, color direction, and contrast combination. A biosemi Act2 multi-channel recording system was used with 64 electrodes over the back half of a 128 electrode cap. This was to increase resolution. As you can see, responses are strongly localized at OZ. Murray et al. 1987 also observed that the cVEP was optimal around OZ. From here on, our data will be focused on OZ. The first stimuli were in the red direction. Here are the waveforms at OZ for 4 different participants, for the uniform square, red-gray checkerboard, and red complement checkerboard. The stimuli were on the screen for half a second, and these responses are the responses within the first 300 milliseconds post-stimulus onset. The different colored responses here correspond to different cone contrasts. The highest contrast is in black, then contrasts decrease from brown and red through the rainbow to violet. While you can see that there is some individual variation among responses from different participants, the waveforms do show similar features. For the uniform square, there is a small early negative peak around 100 milliseconds. The latency of the peak is fairly constant for all the cone contrasts. The red-gray checkerboard seems to show the early peak as well, though the main feature is a much larger later negative peak, which sometimes overlaps with the earlier peak. This large later peak is different in that its latency does not seem to be constant. Instead, it speeds up with increasing contrast. As you can see, the peaks move to the left. In the red complement checkerboard, there is no evidence of an early peak at all. There is just a large later peak that again speeds up as contrast increases. Note that there were fewer stimulus contrasts for the color complement checkerboard due to screen gamut limitations. So the highest peak on the right corresponds to less than half the contrast of the highest peak in the middle. And that these axes are all on the same scale, even so. So we have 2 peaks, an early small one with constant latency, and a later large one with a latency that speeds up with increasing contrast. This suggests the possibility of 2 chromatic mechanisms with differing dynamic properties. We then questioned whether these 2 chromatic mechanisms would be present in both the parvocellular and koniocellular pathways, or perhaps just one. Here I'll focus on the uniform square and color complement checkerboard to isolate these mechanisms and add data for the cardinal color directions. These waveforms are for one participant who viewed all 3 color directions. The top row shows responses for the uniform square, and underneath are responses for the color complement checkerboard. The columns are in the L-M, S, and red directions. Here, the waveforms show 1 full second following stimulus onset. I'm only showing 3 contrasts for each graph so as to make the patterns a little bit easier to see. The highest contrasts are shown in black and the lowest in yellow. Looking at the uniform square waveforms, the early peak is evident in the L-M and red color directions. In the S-cone isolating direction, there is an earlier peak, but it is broader than in the other color directions. For most participants, the S-cone response to the uniform square was weak and in the noise. And in fact, only 2 out of 8 participants showed S-cone responses to the uniform square that were out of the noise, whereas all of their L-M responses were good. So I shall avoid discussing the S-cone responses to the uniform square. A large checkerboard response was evident for all three color directions. The magnitude of the checkerboard cVEP is much larger than for the square. This agrees with Regan 1973, who found that checkerboard cVEP responses were approximately 5 times larger than to a patternless field. It also ties in with there being fewer single opponent neurons in V1 than double opponent neurons. You can also see that there was a sustained response that took the full second to return to baseline. While the square has the early peak around 100 milliseconds, the checkerboard speeds up with increasing contrast for all three color directions. This speeding up agrees with previous work. Excuse me. The difference in the responses to uniform squares and checkerboards is important. It suggests the responses to the two stimuli have different dynamic properties and therefore two different mechanisms, one increasing linearly with cone contrast, the other increase is nonlinear. Here, we show the average amplitude and latency for the L-m and red direction for all the participants. Here, open symbols correspond to the checkerboards and closed symbols to the uniform squares. The red color direction is represented by squares and L-m is the circles. The saturation in amplitude agrees with Rabin et al. 1994. Looking at the latency, the peak latency seems to decay to some asymptotic minimum for the checks, but is relatively constant for the uniform square, again highlighting the two different dynamic mechanisms. There seems to be a shift in latency from L-m to red, where the red has been plotted in terms of the Euclidean distance using the L-m and S axes. If instead we plot the latency as a function of L-m contrast, in other words, the projection of the red cone contrast onto the L-m axis, then the two curves coincide. It looks as if the latency of the non-cardinal color peak is driven by the L-m component. One could possibly think of the latency as filtering out the S component of the stimulus. Summarizing, the cVEPs revealed two mechanisms with different dynamics. One is linear with contrast, and one is nonlinear with contrast. The peaks sped up dramatically with increasing cone contrast. These seem to correspond to single and double opponent processes. And the two mechanisms are present in both the L-m and S cone isolating directions. Now, the presence of a response related to double opponent neurons does tie in with other work that indicated suprathreshold bandpass tuning for humans. Murray et al. Found cVEP results at different spatial frequencies were not consistent with the low-pass model, but they assumed that that was due to some sort of luminance interference. Rabin et al. Reported cVEP peak amplitudes as bandpass for both L-m and S directions. And D'Souza et al. Observed bandpass spatial tuning in human V1 for all three DKL axes using fMRI bold responses. Moving now to measurements of perception, we used saturation ratings. And the main thing to know about these is how we defined saturation to the participants. Specifically, we instructed each participant in the following manner. If the gray background has 0% saturation and a pure color has 100% saturation, what percent saturation would you give to this color? For each participant, stimulus was presented for half a second. Then the participant had to rate what they had just seen. Multiple ratings at each cone contrast were averaged, then normalized with respect to the maximum for each color direction. Then the grand average was taken. Looking at the saturation ratings as a function of contrast, we have one plot per color direction. The checkerboard response is the open symbols, and the filled symbols represent the responses to the uniform square. Here, the spatially averaged contrast is equivalent for the color gray checks and the uniform square. If single opponent neurons are responsible for the perceived color of the checkerboard, then integrating color over the stimulus, the checkerboard and square should be equally colorful. However, the checkerboard responses are rated as more colorful, And note this is for all three color directions. As the contrast increases, the responses move further apart. The additional response must be due to the checkerboard itself, and this implicates a contribution from edge-dependent double opponent neurons that increases with contrast. Therefore, these data seem to suggest that perceived saturation is the combined response of single and double opponent neurons. Now I'm going to attempt to demonstrate this, first in the L-M and then in the S-cone isolating directions. If you are looking at a very small version of this talk, then you probably won't be able to see these effects. Here, both images have the same spatially averaged tone contrast. The dashed lines on the left indicate a central color square which fades to the background. On the right, we have a central colored checkerboard that also fades into the background. If single opponent neurons are responsible for the perceived saturation, one would expect the saturation of the color gray checks to be the same as the color of the blurry patch. However, you should see that the checkerboard on the right is more colorful or vivid than the colored area on the left. This will be the case unless you make the images so small that you can no longer resolve the checks. Only then will they be equally colorful. Here is the S-cone isolating direction. Again, both images have the same spatially averaged cone contrast. So this is also important because it demonstrates how the S-cone responses of the koniocellular pathway also indicate contributions by edge-dependent double opponent neurons. Summarizing, for equally spatially averaged cone contrasts, perceptual responses are larger for the color gray checkerboard than they are for the plain square. This difference increased with cone contrast. It was seen in both the L-M and S-cone isolating directions, as well as in our red direction. Again, if these responses were due only to spatially averaging single opponent neurons, we should have expected the responses for checkerboards and squares to be the same. However, our data showed larger perceptual responses for checkerboards than squares. As the spatially averaged contrast is the same for both stimuli, this perceived increase in saturation must be due to the checkerboard pattern, and this implicates edge-dependent double opponent neurons. Wrapping up, cVEPs indicate two distinct chromatic mechanisms with differing responses to color contrast, one which is linear and one which is nonlinear with contrast. These mechanisms can be isolated using stimuli designed to elicit responses from either single or double opponent neurons. This implies that the two distinct mechanisms correspond to separate contributions of single and double opponent neurons. From our saturation ratings data, we have seen that suprathreshold color perception is more than just a single opponent response. It also involves edge-dependent double opponent neurons. Therefore, we have shown that edge-dependent double opponent neurons contribute to both the cVEP and perceived color saturation of color patterns. While threshold detection is low pass and due to single opponent neurons, suprathreshold color vision is different from threshold. Double opponent cells contribute more to color perception above threshold than at threshold. Now, one explanation might be that as the two different mechanisms have different dependencies on contrast, their relative contributions change with contrast. So while color perception at threshold may be low pass, above threshold, double opponent neurons contribute more to color perception. Therefore, suprathreshold color is pattern dependent. Color and pattern are linked inextricably in human color perception. The combined contribution of low-pass single opponent and band-pass double opponent neurons to perception ties in with the work of Dixon and Shapiro, who suggested that the perception of the dress was based on individual variations of how the visual system extracts low and high spatial frequency color content from the environment. Next, we had similar results for both parvocellular and koniocellular pathways. Given that these pathways are physiologically different, this is quite remarkable. And lastly, Many modern psychophysical papers on color perception use a single stimulus or two half disks as a bipartite field. Given that we seem to have two distinct mechanisms with different dynamics, perhaps it would be interesting in some color studies, especially ones concerning temporal dynamics, if they were separated into those involving single opponent neurons and those involving double opponent neurons. It is even plausible that existing color psychophysical results should be reconceived in terms of these two mechanisms. I will leave you with that thought and say thank you for listening.